Great, thank you, Nick. And our next presenting company is StemCells, Inc. And here to tell us the story is Martin McGlynn, who's the president and CEO.
Thanks, Ted. So let's jump right into it. So I want to talk about StemCells leadership in neural stem cell R&D. A couple of slides here. This highly purified population of human neural stem cells were discovered in late 1999 and published in the year 2000 in PNAS. We have a significant patent portfolio surrounding this investment. We're the first to demonstrate ex vivo expanded human neural stem cells in graft and show properties of true stem cells in vivo. We're the first to demonstrate the potential of human stem cells to treat numerous CNS disorders in animal models. First to treat a human patient in an FDA-authorized clinical trial using ex vivo expanded human neural stem cells. First to demonstrate donor cell engraftment and migration following transplantation into a human patient. First and only U.S. company to conduct neural stem cell transplants into the brain, the spinal cord, and the eye. First to demonstrate, using magnetic resonance imaging, de novo myelin in human patients suffering from a myelination disorder, Pelizaeus-Merzbacher's disease, or PMD, again using human neural stem cells. And first to utilize human neural stem cells in an FDA-authorized trial in spinal cord injury and age-related macular degeneration. So StemCells, Inc. clearly is a leader in neural stem cell R&D. We have safety in 4 clinical trials involving 29 patients. The longest follow-up data we have is over 6 years, and we don't see any adverse events associated with the cells, the immunosuppression regimen, or the transplantation procedure. This year will be the first company to initiate controlled phase 2 clinical trials focused on efficacy with hard clinical endpoints, again using our human neural stem cells. So let me talk about these cells for a moment. They are clearly a first-in-class biologic. This is a highly purified, expandable population of cells. They are stable in cell banks for many, many years. They self-renew, and they do so in a non-tumorigenic fashion. This is an allogeneic process where we take cells from CNS and then we transplant them back into the CNS. In vivo, these cells are hardwired, if you will. They're restricted to the CNS lineage. And we've observed enduring engraftment in the host tissue, biological activity regulated by the host. The cells replicate, they produce more of themselves, and they differentiate in a site-specific fashion into neurons, astrocytes, or oligodendrocytes, again, under the control and regulation of the host. These cells involve many mechanisms, if you will, or modes of action in vivo, because they are regulated by the host, and they respond to the local environment and to the cues that they receive from the host. So we can think of cell replacement using oligodendrocytes, neurons, and astrocytes. We can also consider applications where we're looking at trophic effect. So this is a platform technology where we can take these highly purified biological cells and we can turn them into various products using different formulations, clinical trial procedures, INDs, and in time BLAs. Currently, we are developing applications in white matter disorders. PMD, Pelizaeus-Merzbacher, is one of those disorders. Alzheimer's, where the focus here is on enhanced memory. Spinal cord injury, where the goal is to restore sensation and movement. Dry age-related macular degeneration, where the goal here is to slow down vision loss. And centrally mediated lysosomal storage disorders, where the idea here is to protect host neurons from death. We've observed some dramatic things in the animal world with these cells. They've restored and maintained lost motor function in spinal cord injury. They've protected hippocampal and cortical neurons in lysosomal storage disease animals. We've seen them myelinate denuded, hypomyelinated nerve axons in the shiverer mouse. We've seen them preserve vision in animal models of retinal disorder known as the Royal College of Surgeons, RAT. And work done by Frank Lefferle at UC Irvine in collaboration with StemCells Inc. have been able to restore memory function in 2 different animal models. So truly remarkable observations in the animal world. And of course, the challenge is to replicate some, if not all of that, in the human setting. This is a kind of a schematic of where we are in terms of our clinical development programs. We have completed 2 programs in the brain, lysosomal storage disorder or Batten's disease, Pelizaeus-Merzbacher's disease. Also, we've completed that phase 1/2 study. The results of both studies have been published. And our Alzheimer's program is in preclinical research. In the spinal cord, we're about to complete a thoracic spinal cord injury study. 12 patients. We were actually hoping to transplant the last patient in that study this month, but due to a health issue in the 12th patient, just before transplant, we postponed the surgery. But we will complete that study shortly. And then we plan to initiate a phase 2 proof of concept study in cervical spinal cord injury later this year. And then turning to the eye, we're halfway through a phase 1/2 study in dry age-related macular degeneration. And we plan to complete enrollment this year and open up a phase 2 proof of concept study in that disorder as well. So in the clinic, we have dosed 29 patients, anywhere from 200 million to close to 1 billion cells. To date, we have only reported data on 13 subjects, 3 of which are in spinal cord injury, none in age-related macular degeneration to date. We've got a significant database developing. We've observed patients with NCL for over 6 years, PMD for over 3, spinal cord injury over 2, and age-related macular degeneration over a year now. There are no safety concerns, no inflammatory response post-withdrawal of immunosuppression. We have seen multi-segmental gains in the worst of the worst thoracic spinal cord injury patients. We've evidence of de novo myelination of nerve axons in all 4 of the subjects transplanted with Pelizaeus-Merzbacher's disease. And we've also seen changes in neurological function in 3 of the 4. We've seen evidence of long-term cell survival and migration deep into the brain structure. And we've also seen cells survive for many years post-withdrawal of immunosuppression. All answers to important questions. 2014 pretends to be a transformational year for StemCells Inc. The pace of clinical translation for our company has been slow, but this has been due to the severe restrictions placed on us by regulators as it pertains to first-in-man studies in various diseases and conditions. We've been limited to small, single-center studies, most severely impacted patients, 1 surgeon per study, 1 patient at a time, and long intervals between patient doses. And then finally, long post-transplant observation periods. As we move from phase 1 studies in these conditions into phase 2 proof of concept studies, the landscape is changing, the regulatory perspective is changing, and they are now being more relaxed in response to the data. And so we expect that to result in a significant increase in the pace of clinical translation. So we're embarking on a clinical development program with human neural stem cells, unprecedented in scope, in the doses used, and in the diversity of the indications. We'll be engaging in larger, multi-center controlled, randomized phase 2 proof of concept studies that will have hard clinical endpoints. Our goal here is to get a definitive answer as to whether or not this technology can help the patients that we're targeting. The upcoming clinical trials that I've just described will provide significant insight into the therapeutic potential across a broad range of CNS disorders in the brain, spinal cord, and in the eye. One of the things that we should be looking out for in 2014 is a significant increase in the amount of clinical data that will be made public. We plan to report more clinical data this year than has been generated since the start of the clinical efforts in 2006. It's been a long time coming, but we're getting ready to start releasing more and more data. You will see 3-year data on the PMD subjects in Q2 of this year. We will report out the first data on the dry AMD study conducted here under an IND in the US, and you'll see additional data on spinal cord injury again in Q2 of this year. We also plan to open 20 to 30 clinical sites to support the phase 2 spinal cord injury and AMD trials that I spoke of earlier. We'll be transplanting in 2004 as many patients as we have transplanted since we began the program in 2006, and enrollment will continue to accelerate into 2015. This will generate significant data and insight into the clinical significance of this first-in-class biologic, and we should expect to see that in about 2 years. So it is not 10 years away and always 10 years away, as some wags continue to point out. They say a picture is worth 1,000 words. There are no numbers on this slide except to talk about the years, 2006 when we dosed our first patient in a lysosomal storage disease. We've transplanted about 25 patients through the end of 2013, and we'll transplant about double that amount in the next 2 years. Quick few words on CIRM and Alzheimer's. CIRM has awarded us a $19.3 million forgivable loan to help us engage all of the pre-IND enabling activities for the first ever human neural stem cell transplant in an Alzheimer's patient population. The scientific data suggest that these cells may have utility in Alzheimer's. Others have demonstrated improved cognitive function in Alzheimer's disease mouse models following mouse neural stem cell transplants. Subsequently, neural stem cell derived cells post-transplant have been seen to elevate hippocampal BDNF. This in turn leads to increased synaptic density and restoring hippocampal dependent cognition. We've demonstrated survival of our neural stem cells in the plaque-riddled mouse brain and behavioral and synaptic benefits in Alzheimer's disease mouse models. The findings that we've published demonstrate that neural stem cells can ameliorate complex behavioral deficits associated with widespread Alzheimer's disease pathology. In 2013, as I said, CIRM awarded us a $19.3 million grant. We signed up to deliver an IND within 4 years of that, and our goal is to file that IND 1 year earlier. Quick word on financials. At the end of the year, we ended up with $30.6 million, and at the end of January, approximately the same, which includes the January burn, but also receipt of $3.8 million in CIRM funding. Shares outstanding, approximately 55 million. And now just, if I will, ask for the business for those very rare and scarce investors who might be in the room today. This is a unique platform technology. It has the potential to treat numerous CNS disorders with significant unmet need. We have an extremely well-built and expensive large patent estate protecting our technology. Results of the early clinical trials in multiple indications are reflecting the exciting findings that we have seen in the various animal models that I've shared with you today. We have a rapidly growing body of clinical data. Near-term catalysts include completion of enrollment of phase 1/2A in thoracic spinal cord injury, The last patient will be dosed in the not too distant future in that study. And we will announce additional interim data and start of a phase 2 study. We'll also complete enrollment in the phase 1/2 dry AMD trial, and we will release interim data and start of the phase 2 studies. And I would argue that we have a very attractive valuation for the longer term as clinical data matures. I thank you for your attention, and I'll be outside if anybody has any follow-on questions. Thank you, and thanks, Ted.